Hi, everyone. Thank you for joining today's Health Effects Institute webinar. My name is Yasmin Ramidi, and I am a staff scientist here at HEI. Quick background, HEI is a nonprofit corporation chartered in 1980 as an independent research organization that provides impartial science to inform decisions that foster a healthy environment and better health for all. A quick acknowledgement here that the Energy and Health Program at HEI is funded jointly by government and the oil and natural gas industry and occasionally private foundations. And I would also like to acknowledge all my co-organizers here at HEI and to thank our speakers and moderator for joining us today. Before we dive into it, I'm going to do the usual spiel of a couple housekeeping announcements. So all attendees are muted for the duration of the webinar. You can turn on closed captioning for the event at the bottom of your screen. This is going to be at the right of the Q&A button. We would also request that you submit questions via the Q&A function. We're going to record this webinar and it'll be posted on our website shortly after this. And if you experience any logistical difficulties, please also just send a message to us as the host through the Q&A function as well. Okay, and so a quick bit of background about this project. So HEI's Energy and Health Program is spearheading the design of a cumulative impact assessment for a representative community in the United States experiencing oil and gas extraction and production. So I've listed the objectives of the project here on this slide, but just as a basic summary, the primary purpose of this project is really to help support decision-making about how best to ensure the protection of public health. The design for the cumulative impact assessment is going to incorporate consideration of both chemical and non-chemical stressors from the built, natural, and social environments and how they might benefit or adversely affect health. The project is being overseen by a special panel with diverse experience and expertise that is relevant to this topic. They're listed here. And also with support from HEI's Energy Research Committee. Ultimately, the design will undergo an external peer review and discussion at a public event. This is the third webinar in this multi-part educational series that we've co-organized with HEI's Environmental Justice Program. If you missed any of the prior webinars, you can watch all the recordings on our website. And this being the third one, we're going to highlight more of the methodological aspects related to cumulative impact assessment and cumulative impacts research. We have a wonderful set of speakers today, but due to unforeseen circumstances, Lily Wu will not be joining us. Otherwise, I'm very happy to introduce our moderator, who is Dr. Nicole Diesel. Nicole is an associate professor in environmental health at the Yale School of Public Health. As an environmental exposure scientist, Dr. Diesel's research is focused on applying statistical models, biomonitoring techniques, and environmental measurements to provide comprehensive and quantitative assessments of exposure to traditional and emerging environmental contaminants in population-based studies. Her research uses a combination of large administrative data sets and detailed community-focused studies to advance our understanding of environmental exposures to chemicals, particularly carcinogens and endocrine disruptors. Her contributions have been directed at two main areas, so exposure and human health impacts of unconventional oil and gas development, as well as residential exposure to chemicals in common consumer products and cancer risk, particularly thyroid cancer. In addition, she considers disproportionate burdens of exposures and the combination of environmental and social stressors in the context of her work. She's been a leader in evaluating the exposures, health, and environmental justice impacts of oil and gas development with 30 publications on this topic. She previously served as principal investigator of a large EPA-funded interdisciplinary project entitled Drinking Water Vulnerability and Neonatal Health Outcomes in Relation to Oil and Gas Production in the Appalachian Basin. And recognition of her oil and gas expertise has led to invitations to serve on a public health panel for the California Geologic Energy Management Division, as well as invited testimony before the Senate Budget Committee. So as you can tell, she has a lot of expertise. That was hefty. And with that, I will let you take it away, Nicole.
Hey, Thank you so much for that introduction, Yasmin, and welcome to everyone. I'm very happy to serve as the moderator for this third HEI webinar focused on methodologies in relation to cumulative impact assessments. And it's my great pleasure to now introduce our first speaker, Dr. Nicole Tolvey. Dr. Tolvey is an internationally recognized expert on children's exposures to chemical and non-chemical stressors found in their everyday environments and the influence of these stressors on children's health and well-being. She's currently employed at the U.S. Environmental Protection Agency's Office of Research and Development, ORD, in the Center for Public Health and Environmental Assessment, where she has led several projects that were collaborative efforts with academia, other governmental organizations, and in-house research projects. Dr. Tolvey is the project lead for a cross-ORD project focused on exposures, health, and well-being for selected vulnerable groups, including children. And Dr. Tolvey published the Total Environment Framework, which is an integral component of several recently funded extramural research programs. The Total Environment Framework was adapted for ORD's Cumulative Impacts Report, for which Nicole served as the technical co-lead. Nicole, we look forward to your expertise, and the floor is yours.
Thanks, Nicole. And I want to thank HEI for the invitation to present. I'm going to share my slides. All right, great. Well, welcome everyone. So I'm going to talk to you a little bit today about analyzing chemical and non-chemical stressors for cumulative impacts. So when we think about children and we think about their environment and where children spend their time, it's at home, depending on their age, they could be in school, definitely they spend some time in daycare, and of course they live in the community. And so we know in their everyday environment, they are exposed to many chemical and non-chemical stressors. And these stressors come from the built natural and social environments. Now, as a scientific community, we're very good at measuring chemical stressors, and we have many methods and approaches that have been published by many organizations, including the EPA, to measure both targeted and non-targeted chemicals of potential interest. And of course, there's a really strong body of literature out there with experts who are gathering lived experience, other non-chemical stressors or social determinants of health. But sometimes we don't have those two entities meet, right? And we don't think about the interrelationships between the chemical and the non-chemical stressors. And then not just that complex interrelationship, but then you add in activities and behaviors and inherent characteristics of the individuals, groups, or the community that you're interested in working with. And then you have a big system that you have to understand what's going on and how are all these pieces interacting and affecting their health, well-being, and quality of life. And so that's the Total Environment Framework. It was an organizing framework to get us thinking about how do we make all of these pieces work together, and how do we think about them working together, even if we can't do research in all of these pieces. We have to recognize that they're all there, they're all needed, they're all necessary, and they all have to work together at the end of the day to think about health and well-being at each life stage throughout the life course. So then we used the fundamental tenets of the Total Environment Framework, because it was focused primarily on children when we developed it. But then we said, aha, well, we can actually scale this to actually think about others. We can think about adults, we can think about groups of people, for however we're defining that group of people, whether it's a neighborhood, a geographic location, a specialized occupation, any way you define a group of people. And then you can think about communities. We can still use this construct of the Total Environment Framework to think about the interrelationships between chemical and non-chemical stressors, understanding where they're coming from, who may be exposed to what when, and then taking into consideration also activities and behaviors and people's inherent characteristics. And so that kind of led us to this idea of cumulative impacts and how do we understand cumulative impacts. So in ORD, we have published the ORD Cumulative Impacts Report. We published that in the fall of 22. And it has been widely read all around the globe. And it's important because the definition in there is also shown on this slide, and it's the totality of exposures to combinations of chemical and non-chemical stressors. So it's very much fundamentally routed in exposure science, as well as then the total environment, and trying to think about what are we exposed to at each life stage throughout the life course? How is it affecting our health, well-being, and quality of life? And then, of course, we think about cumulative impact assessment, and that's the process of evaluating how quantitative and qualitative data may be interacting to represent cumulative impacts. And then for the agency, of course, it's related to informing any type of decision that the agency is responsible for, whether that's a research decision or something else. So how do we go about doing this, right? There's lots of ways. This webinar is focused on methodologies and approaches. So in our case, we have a couple of, I'm going to share with you two different approaches that we are using to, or have used, to try to gather information, better understand the whole notion of chemical and non-chemical stressors, and try to think about how this should be working together. So the first thing I want to share with you is a workshop that we completed in the late fall of 2021, where it was focused wholly on non-chemical stressors. And it was thinking about non-chemical stressors within the chemical stressor paradigm. Because remember, the mission of the Environmental Protection Agency is to protect human health and the environment, and ORD's mission in regards to that is providing the foundational science for decisions that allow us to protect human health and the environment. So we're doing not only fundamental research, but applied research, and we're trying to put all these research pieces together. So we had this idea to focus on just non-chemical stressors, but recognizing that they have to be within the construct of the agency's mission. So we invited experts from academia, from nonprofit organizations, and from across EPA to participate in this 2.5-day virtual workshop. Everyone there were probably around 60 people who participated, and everybody was broken into 4 groups. And so those 4 groups, were units that moved to each breakout group. And there were breakout topics on the built environment, natural environment, social environment, and stressors and well-being. And in each one of those discussion groups, and remember, every group moved to every topic area, they were discussing these questions that you see on this screen. So they were asking, which non-chemical stressors should be the focus of the research? What's your rationale for choosing them? What are the criteria for selecting these non-chemical stressors? Which non-chemical stressors are surrogates for others? How should they be managed? And then which combinations co-occur? And so you can imagine there was a lot of dynamic discussion amongst these groups. And then what happened was, after each group spent their 2.5 days discussing, we had folks then summarize all of that information, distill it down, and come up with key takeaways. And so I do want to bring your attention to the link on the bottom of this slide. You can find the non-chemical stressor report there with more information and more of the key takeaways. I just selected a few just in the interest of time and keeping it brief. So what were some of the key takeaways from this workshop in regards to thinking about non-chemical stressors? Environmental protection encompasses both chemical and non-chemical stressors. That kind of seems like a no-brainer, but it's not really something that we, at least at EPA, think directly about because of what the agency's mission is. But we recognize that if you're exposed to a chemical, there'll be a certain biological response. If you're exposed to that chemical and then you have the influence of other non-chemical stressors, your biological response may be different. How so? So we're trying to figure that out. So it does need a systems approach. We do need integrative models, and they're essential for incorporating the non-chemical stressors into the chemical stressor paradigm. Like, they have to be working together, the chemical and non-chemical stressors. All aspects of the built, natural, and social environments need to be considered when we're thinking about how chemical and non-chemical stressors are interacting, what their relationships are, and how they may be influencing health, well-being, and quality of life. The other challenge, of course, is that non-chemical stressors are interrelated and they influence each other. So if you're thinking about a non-chemical stressor from the social environment, it probably very much influences the non-chemical stressors you're thinking about from the built environment. And so then, is it necessary to tease them apart or keep them together? So there's all of those things that need to be thought about. But we did hear from the experts that we do need to think about and appropriately incorporate multiple non-chemical stressors into the chemical stressor paradigm. Like the scientific community needs to move beyond just thinking about one non-chemical stressor, but actually starting to look at, a broad brush of non-chemical stressors. So many non-chemical stressors co-occur. And in this workshop, the group of experts that participated identified these four: geography, neighborhood environment and the characteristics of the neighborhood, housing stock, and racism as the four most important non-chemical stressors that if we can tackle these, we think we can make a difference in understanding the links between chemical and non-chemical stressors. And of course, there was an important message of, approaches and methodologies for analyzing both this quantitative and qualitative data and information are essential. And we need to keep striving forward, even if we don't have everything exactly figured out. We need to keep moving forward and trying to figure out how to analyze all of this information together. So this was one approach that can be used to gather information on a set of stressors. And then the second thing I want to talk to you about is this is a case study example that I'm currently working on with colleagues at EPA, trying to actually analyze using the ideas in the total environment framework and in our cumulative impacts report and what we're learning to try to figure out, can we do a secondary data analysis that makes sense? So this is a very preliminary analysis trying to understand, can we deal with the myriad chemical and non-chemical stressors together to actually look at a particular health outcome? And I want to acknowledge Jacob is a student services contractor assigned to the Office of Research and Development at EPA. So he's been working with Kent and I, and Kent, of course, many of you probably know Kent. He is a senior science and well-known in the human exposure assessment field. So just to remind you again, children are exposed to lots of different stressors, both chemical and non-chemical, everywhere that they spend their time. There has been a large body of work that has shown that both multiple stressors, both individually and in combination, lead to changes in health and well-being. But they're oftentimes not studied except as individual chemicals, or maybe a couple of chemicals, or a couple of chemical and non-chemical stressor combinations. But there's not been this big push to try to look at the big picture of all of the different stressors that may be affecting children's health and well-being. And then if we understand that better, then we can actually scale up to groups and as well as the community. So what was our objective? It was to examine the relationships between the chemical exposure information that was available, social vulnerability factors, and child birth weight. We selected child birth weight as a health outcome to use to show proof of concept. Could we even get this to work? And we're using we're doing a secondary data analysis for the National Children's Study data. We worked with NIH to have access to the data archive. So as I mentioned, we worked with NIH to put in a data use agreement between NIH and EPA to actually access the DASH database where the pilot study data from the National Children's Study is housed. There is a robust and a rich data set of environmental survey and outcome data for the National Children's Study for the different pilot recruitment strategies that they did. So we worked with the Vanguard study. That was one of the specific recruitment types. And then they had a few others. But we started with this one. So we had 518 mother-child pairs included in our analysis, and how we got to that number was they had to have chemical information, they had to have the non-chemical stressor or social determinant of health information, and then the child had to have a health outcome that we were interested in this case, the birth weight. So what we did was, so the chemical information was analyzed either in urine or blood and provided. Some of the samples may have been in dust, and so they were provided directly. And then there's lots of survey information collecting non-chemical stressor information. So our first effort was to try to figure out, how do we put that information together? How do we make sense of it? What do we do with it? So we spent a fair amount of time in the literature, and we came across this publication by Weir, published in 2020, on this PREPARE tool. And the PREPARE stands for Protocol for Responding to and Assessing Patients' Assets, Risks, and Experiences. And it's a nationally representative, standardized tool to organize social determinants of health, to understand a patient's potential risk. And this is administered when people go to the doctor. And it's administered irrespective of why you're at the doctor. You could be in there for a wellness check. They're going to give you this prepare tool to just ask how other things are going. You could be in there for an emergency. They're going to administer this tool. So it's very commonly used throughout the United States in the healthcare setting. And we kind of thought, wow, this might be something that we can use in an environmental setting, like what we're trying to do, to organize all of the non-chemical stressors. Let's see if it'll actually work, because we hadn't come across anything else. So I'll share with you kind of what it looks like after I explained. So we had this prepare tool. The tool organizes the social determinants of health. You can score them in different ways. We decided to score them into 3 groups, a low, medium, and high group, so that we could do a few one-way analysis of variance between the SDOH scores and the chemical measurements, and then the SDOH scores and the birth weight, just to see if we were onto something that might have any level of credibility. So as I mentioned, there were 518 mother-child pairs. This slide shows the participant demographic characteristics for the moms, just based on their race, age, education, income, insurance, and job information that was in some of the survey instruments. And so this is just how how the participants are there. Now, onto this prepare tool. So this slide shows you how we mapped our NCS variables to the prepare social determinant of health factors. So in blue on the screen are the prepare variables as dictated in the Weir publication. And so what they did was they tested this tool, and they did a very robust analysis to try to figure out how the social determinants of health were organizing. And so they came up with these 3 clusters: cluster 1, the social background; cluster 2, social insecurities; cluster 3, insurance and employment. And then they had what they called a standalone cluster. I mean, they probably could have called it cluster 4. They could have called it anything, but they called it a standalone cluster. So we just kept the same nomenclature just to help our brains think this through. And then what we did was, in the yellow, you'll see our NCS. So we went through all of the NCS materials to see if we could find the same information. And you can see in cluster 1, cluster 3, we had an exact match. Cluster 2 and the social insecurities, we did not have an exact match. In fact, you'll see there are many things that just NCS did not capture. But that's the challenge of doing a secondary data analysis, right? Sometimes you can find things and sometimes you can't. And then the standalone cluster was pretty close. And so as you'll see in the blue, there's also a scoring. Each cluster gets a score, then a maximum score of 16. And so we did the scoring, and you can see we have a maximum score of 12. So we thought, okay, let's try it. Let's see what happens. Let's see if we can actually organize all of our non-chemical stressors and actually do any kind of preliminary analysis. So as I mentioned, we looked at the chemicals, and we did selected chemicals. There are a couple of 100 chemicals in the NCS database. We picked several that we thought might be of interest for different reasons to different research groups around ORD. And you can see the chemical analytes here in the first column. And then you can see the number of samples, what the medium is that they were in. It was either urine, blood, except for the lead wipe. And then, of course, as a reminder, when we got finished organizing our social determinants of health, or our non-chemical stressors, we then summed them, scored them, and broke them down. So there were 3 ranges. We had a very low range of 0 to 1. So a range of 0 to 1 means that there are almost no adverse social determinants of health in that person's world. Whereas on the high end, the 5 up to 12, that means they have the most number of adverse social determinants of health in their world. And so that's how it scores. 0 is none, 12 is many. And you can see we broke it down into a range of 3. And then in the p-value, we ran the one-way ANOVAs, p-values. You can see that we had significance for benzophenone 3, PAH metabolites with a couple of the specific PAHs, and then lead wipes. So then as an example, we took the lead wipe data and we plotted it against the social determinants of health factor scores. Again, using the same range breakdown, 0 to 1, 2 to 4, 5 to 12. Remember, 0 to 1 is very few to no adverse social determinants of health in their world, up to 12, which is many. And then, of course, lead wipe on the y-axis is the concentration of lead wipe as measured by the NCS laboratories, with a concentration up to 4 nanograms per square centimeter. And as you can see, as the number of adverse social factors increase, you can see that there is a increase in the concentration of lead in the environment. Interesting. We're not quite sure what it tells us, but it does tell us something. We're convinced of that. Then we looked at the relationship between the social determinants of health and the factor score and birth weight. Remember, we were using this as a proof of concept to say, hey, does this make sense? And the reason we chose birth weight was because there is a wealth of information out there in the peer-reviewed literature. So it kind of gave us a way to check ourselves and make sure what we were seeing or not seeing might make sense based on what other people have published over the decades. And so in this case, we can see that there really is no relationship between social determinants and birth weight. And we were like, okay, well, that's all right. So let me give you a few observations from the work we've done to date. So approximately 15% of the pregnant women had social determinant of health factor scores in the highest, most socially vulnerable category. That was the third category of range of 5 to 12. There were significantly higher exposures observed with increasing SDH factor scores for the PAHs and lead. And then interestingly enough, benzophenone 3, which is a consumer product chemical, was the exact opposite, with significantly lower exposures for increasing SDOH factor scores. So that in and of itself is a very interesting finding. And then there higher SDOH factor scores were associated with lower birth weight for this cohort of mother-child pairs, sort of. So of course, even with this being a preliminary analysis, trying to figure out, does this make sense? Does this have some scientific credibility? Is this something where we actually try to pursue it further? We needed to recognize the limitations in the NCS database. You know, we're doing a secondary data analysis. The data were never collected and processed for us to be able to use them this way. So we're trying to use data that's just available. Also, we have to recognize that it's not representative of the US. Biomarkers were only measured at one time point. Prepare cluster 2, as I mentioned, was really affected by the fact that we couldn't match and align the SDOH factors between what Prepare recommended be in cluster 2 and what we found in the NCS materials. And then we had very few babies in the low birth weight category. But we still think there's actually promise in this approach. So we have some additional next steps that we've been working on the last few months. We're adding in additional chemicals. There are a lot of chemicals in the NCS, and so we're adding in some additional chemicals to think through. We're thinking about other health outcomes. There is a robust amount of information on neurocognitive development and neurocognitive tests in the NCS. And so we're thinking about whether that's potentially an avenue to explore. There's also information to allow for calculations for obesity. We're going to investigate these relationships for SDOH clusters. We want to explore whether we can link and look at joint relationships between the SDOH factor score, the biomarker data, and the birth weight. Because you notice we looked at 2 pieces and tried to look at the relationships. Now we need to put it all together to really drive at that cumulative impact piece. And that's the part we're trying to do now. And then we've actually also taken a step back and we're going back through the NCS data and looking to see, are there other variables that we can use to complete out our clusters? Are there summary variables we can create based on information to complete our clusters? And is there information that we can use? Remember, I mentioned we started with the Vanguard recruitment strategy, but there are other recruitment strategies in the NCS data. And can we use the alternative recruitment strategy to help us actually increase the number of participants that we can then maybe do a more robust analysis, and maybe we would get a better idea of whether this evaluation is actually working. So I shared with you two approaches that we've been using to try to gather information and what we've been up to. And so where's all this work taking us? So I'm trying to take all of these different pieces of information and trying to figure out what methods and approaches should we be using, not using. Are there other methods and approaches that other colleagues are using that maybe we should try and think about? Like, should we think about allostatic load, for example? Should we think about some other stress marker? So those kinds of things. But as I think about that, then I think about the data, and I need to, and I think through, where are we going to get the data? Sometimes it's easy to come by secondary data. Sometimes it's not. If we're going to generate new data, what are we going to generate? Why? How long is that going to take? Do we need it to be raw data? Can it be modeled? How would we go about getting that? So I'm trying to think about those things together, the methods and then the data needs for the potential methods, and how to make that all work. And then lastly, one of the things I'm grappling with is, how do we conduct a real world case study exploring cumulative impacts such that we can then show that the way we're thinking about these interrelationships between chemical and non-chemical stressors really has merit, because at the end of the day, it's how it's affecting people in the real world. And so with that, I thank you for your time. If you want to contact me, my email address is on the slide. So thank you.
Hey, thank you so much, Dr. Tolby, for that excellent presentation. Thank you for sharing your framework and those specific examples. I want to welcome people to put questions for Dr. Tolby in the Q&A function. I see that there are some great questions coming in, so please continue to add your questions for Dr. Tolby, and we will have a Q&A session after the second speaker finishes. So now it's my great pleasure to introduce our second speaker, Dr. Jamie Hart, who is an associate professor at the Channing Division of Network Medicine at Brigham Women's Hospital and Harvard Medical School, and in the Department of Environmental Health at the Harvard T.H. Chan School of Public Health. She is an environmental epidemiologist with a focus on incorporating high-quality exposure measures into ongoing and newly forming prospective cohort studies. She is the scientific director of the Growing Up Today study, probably the best acronym, GUTS, and PI of that project within that cohort to examine the impacts of indoor and outdoor pollution and personal exposures to endocrine disrupting chemicals on semen quality. And she's also been highly involved in expanding the focus of the Nurses' Health Study to include factors related to the external exposome. I think we're going to hear a lot more about those, her exposure assessment methodologies in her presentation. So welcome, Dr. Hart.
Thank you. And like Dr. Tuve, I want to thank Nicole and the Health Effects Institute for the opportunity to speak today, and Dr. Tuve for going first, because it means that she's made my life much easier. So I'll share slides. And so what I'm going to talk about today is going to be a very related topic, talking about this concept of the external exposome. So first, let's see if slides move. One, I just like to define the term. So similar to the total environment, the exposome is this framework or theoretical benchmark that's been floating around since 2005. So for those of you who are thinking about research back in the early 2000s, at the time, a lot of the funding worldwide was going to studies of genetics. So, we were gonna map the human genome, and it was gonna solve all health problems. So, in 2005, Dr. Chris Wild proposed this exposome framework to basically as the totality of human environmental non-genetic exposure from conception onwards. So very analogous to the total environment. And one of the things, I like this graphic from Dr. Martine Verhoeven's group in Europe, because it helps me sort of think through this process, right? Like thinking about everything you've ever exposed to since you were a gamete, is a pretty weighty concept. So in terms of thinking about, how to bin this, right? When, I'm an environmental epidemiologist just by training. So, I've done a lot of studies where I've looked at, the air pollution or traffic exposures with a health outcome, looked at greenness or, natural environments and a health outcome, looked at things like temperature or ultraviolet radiation, right? So a lot of those would sort of be in this circle here, that general external environment. And so again, these are a lot of things that, for example, EPA and other regulatory bodies are able to regulate. In this other top button that they call specific external environment, these are a lot of things that we may have traditionally thought of as sort of just risk factors for disease. So things like tobacco smoking and physical activity and diet and, water consumption. And then consumer products, which here, if you think about them as this specific external environment, the general external environment are things that you have less control over in some ways, right? So you do have some limited control over, your access and where you may be able to live. But the fact that there's a busy roadway by your home, you don't have a lot of control over that. Whereas for things in this specific external environmental bubble, you do have some choice, right? You do have choices in the consumer products that you use in your home and on your person. Again, some of those are constrained by where you live and what's available to you and your socioeconomic background. So thinking about sort of this upstream external environment, this is where a lot of our research is based. However, I just like to be sort of transparent that the bulk of the early work, if you search for, papers on the exposome, a lot of that is more using sort of things like metabolomics or transcriptomics or the epigenome as a marker of all of the things that you're exposed to. So again, really thinking about this as a theoretical framework that's really useful for framing how we're going to do this. In this case, the internal environment is a way to examine how all of the upstream things that you've been exposed to may be interacting. So you'd be able to see the downstream chemicals from as things get metabolized in your body, you get to see the impact of how all these things interact. So maybe that your air pollution exposure is being counteracted by your exposure to green space, a healthy diet, and optimal levels of physical activity. So again, it's a nice way to sort of combine all of these different things. And so initially, this was where a lot of the research was binned, especially with targeted and untargeted metabolomics. Now, again, the thing I always really like about this graphic is, we're always trying to think about how does all of this interact to do health risk and impact assessment, both at the individual level and for exposed communities. So again, thinking through how all of these pieces interact. Now, this is something we've always known. We didn't start thinking that things interacted in 2005, but at the time, there weren't a lot of methodologies that allowed us to sort of incorporate that complexity into our work. Now, the part I really like to focus on, because I think it's where the field is really starting to grow rapidly, is that to be able to conceptualize the exposome, even imperfectly, requires a lot of accurate and reliable exposure assessment in over many decades. So unless you are still a gamete or a very young child, coming up with the life course exposures takes a lot of work. So, in a lot of the cohorts I'm working in, the individuals, could be as old as 90 years old. So that means to be able to fully conceptualize the exposome, I need 90 years of data and maybe 91. So again, that's a pretty high bar. And I think for a while, a lot of the field was a little paralyzed by the search of the perfect in spite of the good. And so I think what you're starting to see now is that people are realizing if you can tackle any of these pieces, you're moving the research forward, and that we don't have to wait till you have 91 years of data to do anything. It's still worthwhile to try. So that's kind of thinking through the framework and how it's very analogous to the total exposure. You'll even see in this bottom circle, things like social capital that again, these social determinants of health, those are also incorporated in this framework because it's something that is not genetic that you're exposed to. So I like to, a lot of times people are like, why is your talk about the external exposome? One, it's just what we do more, but I like to sort of contrast. And I think what I want to say right from the beginning is I don't think one of these is better than the other. And I think actually the most powerful studies are when you have the ability to do both. So if you think about the internal exposome, so that middle graphic, thinking about metabolomics and, epigenetics and those kinds of, and even things like the inflammome, there's a new ohm every day, that or the allostatic load is that those sources can integrate information on the impact of multiple sources of exposure. So for example, if you don't have information on every exposure and every social determinant of health and every traditional risk factor in your population, this may be a place to start because you can get information on the effects of all of those exposures on the individual or community. If you're interested in being able to look at this life course dimension and you have availability of archived samples, for example, childhood blood spots, a lot of cohorts have archived biospecimens, all these different things, you can use those samples to get information on historical exposures. So for example, if you have a cohort or a sample of patients where you have blood samples that have been archived or urine samples that have been archived over time, you can use those to get snapshots across the life course. Again, trying to work towards that full life course framework. Another place where this internal exposome can be very powerful is that it can give you insights into the biological processes impacted by exposures. So again, trying to think about putting that whole pathway together of someone or a community being exposed to a biological or a chemical stressor or a non-chemical stressor, and then sort of what does that look like under the skin? And what sorts, are inflammatory processes being, ramped up? Is it, is it, DNA damage? Again, those kinds of things, thinking about how do we intervene. Now, things that the internal exposome is not typically great at is it can't provide you information on a source responsible for the effects. So if you measure a number of inflammatory markers and look at the inflammome in the internal as your measure of the internal exposome, you don't know if, PM 2.5 or, a number of other chemicals or stress or racism or, material insecurity is driving that measure of the internal exposome. So again, it's a little harder to kind of go back to a source. And unless you have lots of different samples, it's hard to provide information on timing of exposures. So again, are you seeing something in an adult that is the impact of a childhood or in utero exposure, or is it something that they're being exposed to currently? Again, there are ways to get around that, but it requires more data or more samples. So the external exposome also has pros and cons. So one of the things that it can give you is specific information on potential sources and source patterns. So again, if you're thinking about this in a regulatory framework and you want to be able to tie the exposures that someone's being exposed to back, this is one way where you can explicitly do that. It allows for assessment of interactions of sources or exposures on the biological mechanisms and health outcomes. And I'll talk a little bit about that in an example of our work. It provides information that is more readily available into some current policy frameworks. So a lot of policy frameworks, especially in the US and Europe, are based on, okay, we have looked at the literature, we looked at the toxicological evidence, we've looked at exposure assessment, and what we've determined is that there is a level that can be deemed safe for human health for this specific chemical or this specific exposure. And then usually the regulatory framework then acts to go, okay, now we're going to measure that exposure. And if any group defined how, the regulation defines it is above that, then there may be a regulatory action. So again, this sort of single exposure framework that a lot of the literature is based on, this works really well for that. Kind of ties into sort of the older methods, right? Now, where it's not great is it requires information on each exposure source separately. So for example, in the earlier study that we just saw, you needed to have all those blood, urine, and dust wipe samples and be able to measure them. So again, that's expensive. And even when we're using geographic information systems and other modeling techniques to come up with our estimates, that still takes work. Somebody still has to, collect all the raw data, do all the statistical analyses, and then do all of the work to, append that to people in your study or in your communities. And it's more challenging to do historically, again, because you need all of these external sources. So if you did not collect a dust wipe sample 20 years ago in someone's house, unless someone develops a time machine, we're not able to go back and get that data for the external exposome. So again, both of these have strengths and weaknesses. They could be leveraged together to be a more powerful study. So just kind of what does this look like in practice? So I'm going to walk you through a cartoon example of how, in our group, we think about a lot of this. So we have access to a lot of the rich resources of the Nurses' Health Studies and the Growing Up Today study and the Health Professionals Follow-Up Study. And what that looks like on this map, this is every dot on this map is the mailing address of a cohort member. We have about 350,000 right now, over up to 40 years. So you can see that, we have really good coverage, pretty much where people live in the United States. Again, the people in our cohorts we'll talk about are health professionals. So they do tend to live where the people live because that's kind of a fundamental aspect of their jobs. So what you can think about is think for each of those dots. Each dot has a geographic location and a location in time. What we can then do is we work, we develop some of these in-house, and we work with a lot of folks around the world and around the country to come up with prediction surfaces for different exposures. So for example, this is Jeff Yanowski at Penn State, who I did my doctorate with. This was part of his doctoral work and ongoing work to come up with surfaces for particulate matter. So this is specifically looking around the Boston area. So this is just a map of PM10 in this case. But what you can really quickly see is that if you had to guess the sources in his model that are predicting PM, if you know the Boston area, this is 128, this is 495, you can see the Mass Pike. So a lot of these roadways are large sources of exposure, and you can actually see that in the surface. Now then what I do is, this surface is generated monthly. And what I would do is I could take a dot for someone in the Massachusetts area. I could geolocate it on this map, and I grab the prediction for their house for that month. And then I just do that, approximately 4 million more times. So for each, for this, again, this specific exposure is monthly, and we have it from 1988 to up into the mid 2000s. And so again, we just kind of set up a process where for each space and each time we grab the appropriate value for that dot. Now, these dots don't have to be addresses or home addresses or work addresses. They could be GPS points from a cell phone, or from a cell phone application, or from a GPS tracker. So again, we can utilize this framework in a pretty flexible way. So, now, we initially set this whole design up to be able to do this for air pollution. But again, as long as we can come up with reliable estimates in this GIS framework, we can add all kinds of other exposures. So for example, on the left is a model developed by the National Park Service of noise. In the middle is a model developed by one of our former postdocs, Trang Vo Pham, who's now at Fred Hutch, for UV. Down here is a model, satellite-based data of light at night. And in the upper right-hand corner is satellite-based measures of the normalized differential vegetation index, which is just a measure of how much photosynthesis from plants is happening in a given spot. And so you can see here, the eastern half of the US is greener and has more vegetation than the western half of the US. So in practice, we have about 40 of these different types of models where we can append anything to a geographic location. We can also use this in conjunction with measured data. So if, for one of the studies that I'm doing, we have participants wearing these wristbands where we can measure chemicals, we can pair those together. So I can use a geographically based prediction of air pollution with my in-home monitoring data on air pollution, with my wristband-based data on environmental endocrine disrupting chemicals. So you can see how very quickly that you can get a lot of different exposures to start to work with, and also thinking about being able to do this over decades. So just an example. So once we've, been able to have this rich data, and we've been able to look at, these different environmental exposures, one of the great things about the cohorts that we work in is that there are about 100 different outcomes that we can look at, and a whole series of biomarkers. So one of the examples I'm going to use in practice is one of the first times that we, tried to operationalize, even partially this, exposome framework. So the study I'm going to talk about specifically now is the Nurses Health Study 2, or NHS2. It's a prospective cohort study, started in 1989, and as you probably would guess from the name, it is composed of a little over 116,000 female nurses who at the time had a registered nursing license, aged 25 to 42 years at baseline. And what happened, the way that this cohort was enrolled, as you can see, these are the original cohort addresses where people lived at baseline. We had 14 states that were selected because they had large populations and they had registries of all of the individuals who had an RN in their state, and they had some sense of how old they were, and we knew if they were male or female at the time. So they sent a little over 500,000 letters out to the individuals on those lists, and about 116,000 responded. Now, what's really great for our research is that as part of the normal follow-up of this cohort, the participants receive a biennial questionnaire every 2 years to collect a lot of information on health and lifestyle factors. And luckily for us, even now, the questionnaires are mailed, which provides us a residential address history. So going from the original baseline locations of the cohort, individuals have moved so that we have at least 15 nurses in this cohort in every state. And now, even when participants are doing the questionnaires online, for the most part, we do still keep a record of their address histories updated at all times. So what you can see here is that these are the years in which the questionnaires are being asked. And so we have detailed health and covariate information for all of these years, and we have the addresses, which we can then use to append a bunch of exposures. Now, the other great thing about being in a, an ecosystem with, this many other researchers is that, as soon as someone else contributes a resource to the cohort, it's available for use. So, for example, we had this doctoral student, Hoi-Chu Lee, who her whole interest in her thesis was looking at age at menopause and the menopausal transition. And so her earlier thesis papers were on a whole host of different environmental exposures and those questions. And then for her final thesis paper, she wanted to move a little more inside. So thinking back to that diagram I presented, really looking at how external factors were impacting the internal environment. And here she just chose a single biomarker, which is anti-Müllerian hormone, which is a marker of ovarian reserve. So sort of how much ovarian reserve is left in a woman as they age. So what she had information from is from other studies. There were almost 2,500 women who had this measured in a blood sample. And this again was available. It was from a series of case control studies. So that's something important to think about. They were most of the participants were 40 years of age at the blood draw. So in all the cohorts we're working in, there are blood samples collected pre-disease for a lot of different participants. And then what's happened over time is as people have wanted to look at different diseases, you can pull those earlier blood samples to look at measures of future health out risk. So she was able to do this, and so all of her participants were premenopausal. And then what she did is she took a slightly more limited approach. So not the traditional XWAS where you're looking at 200, 300, 400 different environmental exposures. She looked at a series that had been shown to have some relationship with either menopausal status or this hormone specifically. And so what she looked at was she took cumulative average exposures. So from the beginning of the cohort till the time of providing that blood sample of a number of exposures. So greenness, 2 measures of air pollution, PM2.5 and coarse PM. A few measures of noise, light at night, ultraviolet radiation, and a series of hazardous air pollutants that have been shown to have endocrine disrupting features. So for all of those, again, we appended those to all the addresses, and then she took the cumulative average until blood draw. So just to walk you through, again, don't get intimidated by the statistical methods here, but just to think about how we would walk through this. So what she first did is what traditionally has done in the literature in hundreds of papers is take a single exposure and look at its association with this biomarker. Then we scaled up and she looked at multiple exposure models. And then to see how things group together, she used this technique called principal component analysis that essentially just clusters together the different exposures, how they relate. So if you could think about how correlated different things are, those are going to group together. And then she used one of these new methods that have been designed for mixtures analysis called hierarchical Bayesian kernel machine regression, or much easily confirmed as BKMR. And what that method and similar methods are able to do is to be able to take all these highly correlated exposures. So again, we don't expect these things to be occurring randomly. We expect that they're coming from different sources and that those mean things are going to cluster together. These methods and similar methods are able to account for that correlation and utilize it to give you a sense of how the overall burden of exposures are impacting what you're looking at. She was able, again, because we have all that rich questionnaire data, to adjust for a number of confounders that have been shown in these cohorts to predict levels of this biomarker. And then this is what she's able to do. So again, just thinking about when you have all these different exposures that, traditional methods where you would just put them all in the model don't work well because of the high correlation. So if you look at this graph where blue circles headed this way or blue ovals tilted towards the upper left corner are negative correlations, and the paler color and red leaning towards the upper right corner are positive associations. So what you can see is, not surprisingly, things like PM2.5 and PM10 are correlated with each other, and they're correlated in a negative direction with vegetation and things like that. So a lot of these patterns we see repeatedly across different cohorts, and just the strength varies. But again, we're looking at correlations, in some cases up as high as 0.8 or negative 0.8. So that makes original methods challenging. These are a lot of numbers, but what I just really want to point out is that when we looked at these single exposure models, again, you want higher levels of AMH to some degree, because that indicates that you have higher levels of ovarian reserve. So in single exposure models where we're just looking at one at a time, increased vegetation around your house is associated with, it's positively associated. So you're seeing higher levels of AMH, again, not statistically significant. And when you're looking at sort of the negative associations, the strongest one is formaldehyde. So as you get increasing exposures to formaldehyde as measured by the EPA NADA databases, we see that you have lower levels of AMH, or so lower levels of potential future fertility. Now, the thing that a biostatistician would tell you should never do is in this far out column, she put everything in at the same time. And as you would expect, and as if you ever took a biostatistics class, our confidence intervals get really wide because of the high correlation. And the really thing we only see hang on is that formaldehyde analysis. So again, we were doing these initial steps just to be able to compare to some other papers in the literature that had done single exposures, and to give us a sense of the directionality that we should expect in our more complicated models. Because if you start with the more complicated models and they're hard to explain, you're kind of in a bad position. I talked about that she did these principal component analyses. So again, what we're looking at here, sort of how things cluster together. And so this is just a graphic representation of how different things are correlating together. So we ended up with 4 separate components. The first one was light at night and a whole bunch of those potentially endocrine disrupting chemicals. The second was NDVI, coarse air pollution, and UV. The third was noise, and the fourth was PM2.5. And what you can see is if you look at the impact of each of these components, so increasing all of the exposures in this component on AMH, that, sort of chemical exposure 1 is leading to negative changes in the AMH. Everything else is a little messy, and PM2.5 is in this model showing positive but more wide confidence intervals. So really, part of the reason we did this step to look at the clustering was to be able to see how we would expect things to cluster together in this more complicated BKMR framework. So what I want everyone to look at first is just B. So what BKMR is trying to do is if you increase each of the exposures in the mixture you're looking at, so for in this, we were looking at about 15 exposures. If you increase each by 1 decile, so 10%, what do you see? So here, compared to the median or the 50th percentile, what you see is that as the whole mixture increases, the AMH levels go down. So the way you would interpret that is that overall, this mixture of exposures is harmful. So it's leading to lower levels of AMH. What you're seeing in panel A is some of the exposures that are driving this overall mixture association. So that green vegetation, which we were seeing some complicated results with when we were looking at it just linearly, that's because it turns out it's nonlinear. So you see benefits up to a certain point, and then it kind of levels out. Whereas benzene and formaldehyde, you're seeing this negative slope, meaning that for each, as you can go across the range of benzene, that's leading to higher levels, lower levels of AMH. So again, this is just a way to really think about this holistically. You can increase the number of exposures. You don't have to have all chemicals. Some of the work that we're working on, we're putting in both chemical and non-chemical stressors into these types of models to be able to see how they both interact and how overall the weight of a burden of exposures for either a community or a person, what does that look like in terms of overall health? So just to give you a sense of the other kind, so that was just one example using a biomarker. Part of the Nurses Health Study 2 is that Growing Up Today study, or the GUTS cohort. Those are the children of the Nurses Health Study 2. So for a large amount of those individuals who are now in their 30s and 40s, we can actually look at their entire life course because their mothers were enrolled in the cohort from the time of their birth, and now we have data on them, throughout childhood and throughout adulthood. So there we're able to look at the impact of intergenerational exposures and birth outcomes in the children. We're also looking at things like how do things like untargeted metabolomics mediate association. So is it on the pathway between these environmental exposures and mental health? I hinted that, in addition to looking at addresses, you could look at GPS locations. So we're doing a lot of work to see how multiple environmental exposures, both chemical, non-chemical, impact physical activity and sleep that can be measured using wearables and GPS locations. Looking at multiple environmental exposures and risk, and also looking at things like prostate tumor histochemical markers to see how these environmental exposures and non-chemical stressors and social determinants of health may be impacting the chemical environment in a tumor that is predictive of outcomes from cancer. So just to wrap that all up, the way I like to think about this is the exposome is a useful framework for examining the impact of multiple environmental exposures across decades on myriad health outcomes. There are lots of similar frameworks, so I don't think you have to be team total environment, team exposome. They're all getting at around the same kinds of things, just with some slight differences in methodology. But I really also want to emphasize that this is building on decades of single exposure, single outcome environmental work, and that a lot of this required technological advances in several areas to be able to do this multi-exposure framework, especially on the biostatistical and data science side. And I think one thing that will be interesting to talk about a little bit in the Q&A is how do we take this information and make it useful for, cumulative impact assessment, risk assessment, because again, some of those frameworks were originally developed for single exposure, single outcome models. And I think it's really an exciting time to be able to see how can you look at the totality of exposures for an individual and a community and use that for regulatory purposes. I mean, we do it for things like blood lead, where, we don't necessarily, when a child is getting their blood lead measured, it's not being measured to detect a certain source. It's to see if the child has a problematic or concerning level of blood lead. And then there's a lot of work to figure out the exposures. So again, we do have some frameworks for this, but I think it's interesting to see where things are going to go. You can expect that, when that many exposures need to be generated, it takes a really large team. So here is my current team. The QR code should take us to the website. I may put the link in the chat as well, because the Harvard School of Public Health is in the middle of redesigning their website. And so the links sometimes are not working all that well. And I just want to say thank you again, and happy to answer questions during the Q&A, or if you have questions, you can send me an email.
Hey, fantastic. Thank you, Dr. Hart. Another excellent presentation. And again, please share your questions in the Q&A function. We have some great questions in there. I'm going to kind of combine some of these questions and put my own spin on them. And I'm going to start a little bit bigger picture and then get down to some more specifics if we have some time. But I thought I would start out first just with language. And Dr. Hart, you kind of alluded to this in your last slide. Both speakers, presented these different frameworks. We have the total environment framework, we have the exposome, cumulative impacts. Sometimes language and terminology can be a uniting factor. Sometimes it can be sort of a barrier to, collaborating and things. But I'm just wondering if each of you, and maybe we can start with Dr. Hart since she just mentioned this, but how are you thinking about, the exposome versus these other kind of frameworks? And are they how are they related, or in what situation might one be better than the other, or how can we learn from these different frameworks?
No, I think it sometimes is a deep source of frustration because, there are some groups where exposome means a very specific thing and it means only untargeted metabolomics. And so in those spaces, I don't use that term because that's not how I mean it. Sometimes we go instead with multiple exposures across the life course. It just doesn't roll off the tongue quite as nicely. So I think it is an interesting thing that there are some connotations with all of these different terminologies that in different spaces mean different things. And so I always like to start out with really clarifying when I'm saying this word, this is what I mean, because I do think that sometimes it becomes a barrier because people are like, oh, that means you're doing only this lab-based stuff and it's too complicated and that's not what I mean. And so I do think it's useful. They're useful frameworks, but yeah, the terminology people are getting a little caught up, I think, in sometimes, and there's some camps, which.
Dr. Tolvey, do you want to add to that, how you're thinking about how these concepts kind of relate?
Sure. Yeah. So, it's funny that you should ask that question because we were challenged with actually trying to describe how the total environment framework and the exposome link together. And I felt like I was, excuse me, like I was defending my dissertation, but there is an awful lot of overlap. And I think some of it is terminology, some of it is like the sector you're working in sometimes, but I think at the end of the day, the total environment framework allows the flexibility to not have as much, like the exposome has more details in it and people add lots of pockets, like you can see different figures of the exposome with different circles where they're adding a lot more detail, whereas the total environment framework is getting the way it's constructed with those 4 circles in the one ring. That is actually a very specific type of diagram that shows that they are all interrelated and they have to be working together to get to the center ring, which is addressing health, well-being, and quality of life. So, but we're not trying to take it that much further other than to get people to think about that construct, and then the people, right? Whatever objective you're trying to address, you need to fill in the pieces to help you actually use the framework to get there. And I want to say something about the cumulative impact. So the total environment framework built to the cumulative impacts. One of the biggest things about the cumulative impact framework and cumulative impact assessment is there's a strong community component, which is not always evident in these other pieces. In fact, it may be completely absent in the other pieces, but cumulative impacts, a fundamental tenet of if you're actually doing cumulative impacts, is you engage the community in your work.
Wow. Great. Well, why don't I follow up on that? Do you have a specific instance in which the community was engaged in, one of your examples? And maybe you can expand a little more on what community engagement would bring, what value add that would have to these frameworks?
Sure. So as a disclaimer, not to avoid the question, but to give it context, I'm in the Office of Research and Development. So we oftentimes do not work directly with the communities, but we work through our regional partners. So the EPA is the United States is divided into 10 regions. So it's our regional partners that are the boots on the ground interfacing directly with the communities. And there actually are several examples of where they have been attempting to do cumulative impact work in support of communities. I can direct you to the EPA website where there's some examples for the Chicago area, the Dallas area. I think those are region 5 and 6, region 2. So there are actually activities going on all around the country in regards to trying to do the cumulative impacts. And we provide a lot of support. Like we bring the science expertise to the regional folks so that they can actually try to implement things in real time and in a real application. And the second part of your question, Nicole, was one of the important pieces is the lived experience of the communities, capturing that lived experience component, which we don't capture well right from in the research, because we're buried in the research and we're not thinking about that piece of it.
Okay, great. Thanks for those comments. Moving on to kind of decision making, I guess there's some good questions in the chat about whether you can share if there's a specific example where these approaches are being used to inform decisions, or if not, what additional advances would be needed so that these are ready to inform decisions. And one of our listeners asked, specifically, this seems like this could be quite challenging because some of the different exposures cross regulatory authorities, like EPA doesn't, they said in their words, doesn't control racism, but you could see that there could be, light at night versus noise pollution, the food environment could all cover different agencies. So thoughts about how these tools could be used to inform regulatory decisions?
Yeah, I think it's something we struggle with because we always want our research to be as policy useful as possible. And I think that's the exact challenge, is that when we're mixing hazardous chemicals and structural factors and housing and, poverty and all these things together, we are very aware that those are not, that is what the community is experiencing, but that is not a clean regulatory, thing. So it is somewhat part of our approach is also providing these single exposures when we're doing this study, because in some cases that is more useful for different, bodies to work with. And then knowing how all these interactions are happening is useful, but is often kind of thought about in a, let's look at the most susceptible populations when we're developing a regulation. So, if they know that there are interactions between maybe a chemical exposure and some of these social determinants of health, that helps in that regulatory piece. But I think it's something we struggle with a lot when we're doing these, I think we just did one the other day when we have like 25 different exposures that would all be regulated by someone different. And thinking, talking with students about like, how do you present that in a way that is useful to anyone except scientists?
Yeah. Nicole, I know you're in the research arm, but you are in a regulatory agency. Do you have other thoughts about how these, and you said that the goal ultimately is to inform decision-making. So thoughts about how that interface will happen and how close are we to being able to use this to inform and support decision-making?
Yeah, it's the one that question is there's a lot of conversation going on around that question in the agency and all different parts of the agency, because of course there's all the statutory authorities, and depending on the way those statutory authorities are enforced depends on whether there is a opportunity to actually consider cumulative impacts. I'm not sure where folks are at, since I sit in the research realm and help support the decisions, but I'm not sure if anyone has actually made a decision yet based on using truly cumulative impacts. I mean, one of the things we did note in our ORD report, on the cumulative impacts report, is that we the federal family should not shy away from, yes, okay, EPA, our mission is to protect human health and the environment by regulating on chemicals. But if we're doing some cumulative impact work and we find something that's, related to HUD, well, how do we reach HUD, right? And get them engaged in the discussion? How do we reach, FDA, USDA, whoever it is, how do we bring the federal partners together to address that community need? Because exactly what Dr. Hart said, there's all these different parts that need to come together and work together. That's a big lift, but I mean, it is the challenge in front of us that we're really trying to do. Have we been successful yet? No, I can't think of a straight up example.
Okay. Let's turn to some questions about some of the methods. For example, this question is for both of you. You both presented these kind of tools and models and methods for kind of organizing these different stressors. One question is, I guess, how to identify which stressors to include. So, for example, someone asked, are you going to be looking at microplastics, Dr. Tolvey, or other metals? I think Dr. Hart, you just said you had like 41 different exposures. So, I guess how to identify which ones go in, and also how to you're organizing them now, but are we weighting them differently, or any thoughts about when we combine them, how to prioritize one type of stressor over another? Who wants to go first?
I can tackle, add a few comments first. My mute button's not working very well, so I'm slow on the trigger here. But so the question is a really good one, right? And so if we're really in the cumulative impacts realm, remember, one of the base elements of true cumulative impacts research and work is that you're getting feedback from the community. So if the community believes that lead is the primary issue that they are most concerned with, if they believe it's microplastics, if they believe it's garbage and garbage collection, or the dump that's down the street and all the trucks need to drive through the dump to get, or, you know, through their neighborhood to get to it, you know, so it's getting the community's perspective and incorporating and building the solutions around the community's perspective on what they need. So it could be any of those and other things, combinations of things. It could even be, you know, as a researcher, you know, we think we're going in with X agenda, and, you know, that may not even be the community's agenda. The community's agenda is A. And so then we have to figure out how to actually be responsive to what the community needs, you know, so yeah, yes, to a lot of that.
Yeah, and I think we struggle in a different way because, you know, our community, when we're using these cohorts, is, you know, across the entire US. So there's no geographic sort of way that they're hung together, but they have this identity as participants in these studies. And so, you know, for us, a lot of times environment is not maybe even in the top 10 of things they would be interested in us looking at. They're worried about job stress and, you know, burnout and things like that these days. So, you know, we're always trying, especially on the questionnaires, to frame that. But I think more directly for the environmental exposures, sometimes it's really that there are exposures we desperately would love to look at, but we can't reliably either measure historically or measure for the entire continent. So noise, I showed that National Park Service model, it's time invariant. Every time we use it, we desperately wish we had a measure that, you know, was more like what they have in Europe, and, you know, time varied and things like that. So I think it is a real, like, so, but our philosophy in thinking about exposures is we want it to have sort of a plausible biological mechanism and be something we can measure. So there are definitely lots of things that are on the table that we've wanted to look at and can't. And there are things we could look at and don't think it's, you know, we don't think, and, you know, from talking to our participants, they don't think we should really look at. And so that's just the way we approach it a little differently, not working directly with, you know, like a geographically located community.
Right, that was a really nice illustration of some of the differences when working on different scales, you know, working so that this framework could be flexible, like in Dr. Tolby's scenario, which is describing to, you know, be flexible to prioritize based on different communities versus Dr. Hart doing these national studies, and there might be other factors. So I just want to pick up on this theme of scale, because there were a couple questions about that. So Dr. Tolby, you know, there was a great question about sort of what's the end goal of your research? Would it be to apply this framework to certain like narrow geographic areas, or something else, you know, like census block levels or other things like that? So how are you thinking about that issue of scale in terms of kind of the output of this research?
So for us, we are grappling with how do you take local information, so at a community neighborhood scale, and how do you scale it up to national? Like that is our grand challenge in front of us, is how do we make it more applicable broadly?
Great. And then Dr. Hart, there's a question. You're kind of working kind of the opposite. So could you speak to this issue? Someone asked, can you talk about issues of scale when you're using this broad environmental data and then ascribing it to the individual nurses in your health study?
Yeah, and so I think, you know, for us, we're always trying to get measures that, you know, are at a very fine geographic scale. So like there are multiple air pollution models floating around in the world. We use one that we can predict at an address versus on a grid, just because we have that data. But I think in terms of like being able to use the geographic information of our participants to advise a community, we don't tend to do that, because then we would have to also make the claim that our participants are living in geographically representative locations in that community. So when we're looking at sort of a community context, what we'll do instead is, instead of just assigning those exposures that we have to points, we may, you know, expose, you know, zip codes or, you know, the centroids of census things to be able to look at, you know, we've looked at some things like looking at interactions between, you know, poverty and different racial and ethnic groups and their exposures. So you can use those same frameworks to get information on communities, but we don't do it going through the addresses of the cohorts, because we're never comfortable to make the claim that they're representative of any given neighborhood that they live in.
Great. And following up on that, there's a question in the Q&A from Andrew Geller, and I think it's sort of related to this area, oops. Actually, do you have it up there, Jamie? You could.
Sorry, I just made it go away. I think I half answered it when we did it.
We did. Okay. Is there anything else you want to say about that one? Okay. Okay, great. Okay. And maybe both of you could talk about a little bit more about cumulative impacts. People have some questions about the technical terms, like, are we adding things together, or Dr. Hart, you showed there were some instances where things are kind of going in different directions. So can you maybe talk about what we mean by cumulative and how we address when things are going in different directions?
Yeah, so I think one, I saw the different directions was, I think, things flipping signs and single to multi exposure models. That was really just a fact of in both cases, those were null, but flipped sign when we put everything in together. So I just wanted to address that specifically. But I think in terms of thinking of the cumulative, you could be very direct, and some of these methods will give you a number of the mixture that you've put in, right? So like, that's one way of saying, like, this is the cumulative impact of this mixture of exposures. And depending on the method you're using, their interactions and their nonlinearity. So depending on how complicated you get. So that's one way of doing it. That is not usually what's meant by like cumulative impact. It's just I do not even pretend to, take anything more than my one risk assessment class too far. But, thinking about, that's what I was talking about with the cumulative effect is all of the exposures that we're looking at together. And these new methodologies can give you the impact of all of those together, and also give you a sense of what's driving, which individual factors or interactions are driving that. But that's not the same.
And Dr. Tolby, do you want to add to that discussion?
Sure. So I think as Jamie mentioned, the cumulative impacts is the totality of exposures to all kinds of things, right? All throughout your life, and what's happening there at different stages with your health and well-being. I guess we should never always think about it in the negative sense, right? Like, it's always a decrease or a decline, or because there's so much evidence in the peer-reviewed literature that green space and park space is beneficial. And so it's like, how do you take that into account? And then how do you take into account the resiliency of the system, whether it's individuals or the community, to bounce back from whatever's being thrown at it, so to speak. So all of that needs to come into play to kind of figure out what is really going on with, people in the community. And I don't think we have like a straight up answer, right? Oh, it's always going to be additive. Or if you have X and Y stressor, it'll be additive. But if you have Y and Z stressor, it'll be, synergistic and negative. I don't think we're there yet. And I don't think we can figure that out at this point. Yeah.
Okay, great. As we start to wind down, I want to ask the speakers to kind of start looking to the future. I guess, are there other methods we haven't discussed that are ready or close to being ready in this space? Some other things that sort of came to mind during the presentations was, we have things like the EPA EJ screen or the EQI. Are these tools that could be used in this space, or are there other tools or approaches available that we haven't covered yet in these presentations?
Okay, I'll go first. Yeah, so I do think there are probably a lot of screening tools, right? You mentioned DJ Screen, Cal EPA has Cal Enviro Screen. I do think there are tools out there that can be used as screens to help us figure out where maybe we should be spending our time, as a scientific community, who's more impacted than others. Not every community is created equal, and we have to figure out how to help everybody. So how do we do that, and how do we do that efficiently as a scientific community? I do think there could be methodologies and approaches that are maybe closer than others if we're not looking It depends on what we're looking for. Like if we're looking for just people are stressed, what's your baseline level of stress? If there's a perturbation in the system, how does stress change? Then all we have to do is measure stress, right? Measure a stress biomarker in a biological system, and we would kind of have that answer. Is that enough? I guess as a scientific community, we have to ask ourselves, is that enough information? Is that all we need to know? Or do we need to know specifics? Do we need to know what the stressors are that people are experiencing? You know, I've been doing this research for the last several years, and I keep asking myself that, do I need to spend all this time trying to figure out and understand the individual stressors, or is it just enough to know that people are stressed and that communities are stressed, and then how resilient are they? How do they bounce back? I don't think there's any easy answers. You know, I did in my remarks, I did mention, you know, the allostatic load. I think that's something that maybe for just a human system, maybe that will work. Lost my train of thought. In the eco world, there are different ways that you can look at natural indicators. Is that enough to understand, you know, the health of the ecosystem? So, you know, I do think there are some tools out there that folks are using, and there are some methods and approaches. The question is, will they work for this grand idea of looking at cumulative impacts?
Okay.
Yeah, and I think one thing that, you know, I think was really helpful early on that you said is, I think the where I kind of, everybody's trying to challenge the big piece, and I think a lot of where the movement is people solving small parts of the puzzle and putting it together. I think that's where a lot of like what I see the very useful stuff is happening, because again, yes, I can build a crazy giant model that no one can understand the output of, but like that may feel satisfying that I, you know, was able to get that model to run on a computer, but like, what does it mean? And so I think that's where some of the challenge is that like some of these technological advances have allowed us to ask more complicated questions, but sometimes we don't do the pre-thinking to ask those complicated questions. And so I really think, you know, grounding it in kind of, you know, like community, like even if you're not working with a community, thinking about how this applies to a community is always really important, because you don't want to tell somebody something about themselves and have no idea what you're talking about, which I think sometimes with some of these more technological methods we do a lot of. So I think that's where some of the work needs to happen too, is really in thinking about, yes, machine learning is cool and fun and we're going to get some really great insights from it, but are they useful insights, and are they insights that are going to be applicable to either a regulator or a community member who's just trying to figure out like how to keep their family safe? And so I think that's where some work also needs to happen, is kind of maybe even just thinking about empathy and compassion in your modeling.
Okay. What a great note to end on. And I want to thank both of our speakers for these really rich presentations with so much expertise and insights. And I think I will turn it over to Yasmin for final closing remarks, and thank everybody also for attending this webinar.
Yes, I lost my slide. Okay. Well, I just wanted to say, you know, thank you for attending this webinar by HEI on cumulative impacts. The webinar is going to be available on the website in a couple of weeks. If you want to continue to follow the activities that are related to the overall project, you can sign up for updates on the Energy and Health website. And then also we're going to send out a feedback survey by email right after this webinar. So we, you know, please take the time to complete that, and we appreciate your feedback and hope you will, you know, stay in touch with the project.